Okay, so hi everyone. Welcome to another session of Sybeco. We have Abby Silbaugh present her work, her recent publication today. Abby is a neurobiology graduate student in Christian Hansel's lab at the University of Chicago. She's broadly interested in the olivocerebellar system and how it interacts with the rest of the brain. She's going to present her work on how cerebellar climbing fibers gate plasticity in somatosensory cortex via modulation of neocortical interneurons. She also just recently defended her thesis. So congratulations, Abby, and thanks for taking our invitation.
Thank you. And thank you everyone for coming. Feel free to ask questions during this. I'm happy to answer them. So the title of this talk is Cerebellum Gates Plasticity of Primary Somatosensory Cortex. And scientists have been studying the structure and function of the brain for thousands of years. And one of the first observations made by the early anatomists is that it seems like the brain is divided into two parts, the big brain and the little brain, otherwise known as the cerebrum and cerebellum. And the cerebellum is an evolutionarily conserved structure, meaning there's a cerebellum or cerebellum-like system across species. So pick your favorite animal, and if it's not here, I guarantee you it probably has a cerebellum. And sadly, it's been studied in isolation for centuries, which really limits what we know about its function. What do we know? For as long as we've been studying the physiology of the brain, we've known that the cerebellum is involved in motor control. So this guy, Luigi Rolando, who was perhaps the first to complete a set of experiments that established the function of the cerebellum, wrote in 1809, "I've consistently observed that the loss of movement was proportional to the extent of the injury inflicted upon it." And this is still very true. However, recent studies have shown that the cerebellum has many roles outside motor control, including in language function, sensory discrimination and perception, and even social behavior. And these behaviors might seem very different, but the cerebellum might have a ubiquitous role in all of them. So let me give you some specific examples to show this. So starting with motor control, the most classic example, we know it's involved in coordination. So if you're reaching for a target and you go off course, the cerebellum is activated so that corrective movements can be made, both while you're reaching and the next time you reach. So in this way, the cerebellum corrects the error in reach trajectory. We also know that the cerebellum is involved in processing semantics, so it'll become activated if there's a difference from what is expected. So for example, if you start reading the sentence 2 plus 2 is, you might predict it'll say 4, and if it instead says apple, the cerebellum will be activated. So it recognizes this error, here a mismatch between a predicted word and the actual word. And similarly, for sensory discrimination, a difference from expectation in a sequence of sensory stimuli will cause the cerebellum to be active. So perhaps you're exposed to a very regular stimulus like this one, you'll begin to predict that the next stimulus will be similar to what you just experienced. And if instead some quality of the stimulus is different, the cerebellum will be activated. And finally, even for social behavior, a violation of a social norm might activate the cerebellum. So you'd predict that someone approaching caution tape might turn away, and if instead they break the rule and decide to cross it, evidence has shown that the cerebellum will be active. So in all of these examples, the cerebellum seems to be activated when then there's some sort of unexpected event, mismatch, or put really simply, an error. And it makes a lot of sense that the cerebellum shows broad activity in these situations because the cerebellum itself is an error correction machine. Many of you are probably pretty familiar with this, but I'll give a brief overview anyway of how this works. So Purkinje cells are the main computational unit of the cerebellar cortex and are able to sample sensory and contextual information from hundreds of thousands of parallel fiber inputs from the periphery and the rest of the brain. And when there's an error, climbing fibers become active and signal that the current output or prediction is incorrect. So this climbing fiber input causes plasticity of the coactive parallel fiber inputs, updating the Purkinje cell's future responses. And as a result, the next time the same situation occurs, the cerebellum's output is adjusted to correct the error. So in many forms of motor learning, this climbing fiber-driven plasticity in the cerebellum is required. And in this way, the cerebellum is considered to be the storage site of a memory trace for learning. However, the cerebellum isn't the only brain region involved in these examples that I've just given. The cerebellum has extensive reciprocal connections to other brain regions and interacts with them to give rise to these more complex behaviors. So in similar scenarios to these that I've described, we know that neurons in other parts of the brain respond to error, mismatch, unexpected events, and so on. And we know that error-driven plasticity can occur in other parts of the brain. So where are these error signals coming from in other parts of the brain? What could be driving this plasticity? And what if it's the cerebellum and cerebellar climbing fibers in particular? So these are kind of the questions that I was thinking about in the beginning of this project. So while this might seem plausible, we don't really know if this is true because the role of climbing fibers outside the olivocerebellar system has never really been shown. So this brings me to my central question. Can cerebellar climbing fibers influence the activity and plasticity outside the cerebellar system? And so why has this never been studied, even though the cerebellum generally seems to be active when there are errors or situations when the climbing fiber is activated? Just looking at a very quick PubMed search, looking at all the papers that include the word cerebell or like cerebellar, cerebellum, we can actually see that human studies have outnumbered animal studies in recent years. And this is even when you include cerebellar stimulation. And I find this pretty interesting because a lot of these human studies are really pushing forward what we know about the cerebellum outside motor control. Yet we don't have a lot of resolution with these types of studies to really understand how the cerebellum might be interacting with other brain regions. So only recently have studies looked at this in more detail. Perhaps in the last 5 years, have we had like laminar specificity with our recordings. Some people have identified putative different cell types in the cortex. But really, in all of these animal studies, still people have been modulating the outputs of the cerebellum, not climbing fibers themselves. So we wanted to really understand, does the climbing fiber have any role outside the cerebellum? And in particular, how is it modulating the different cell types in the neocortex? So back to the central question, not only can they influence activity and plasticity outside the cerebellum, but if so, how are they doing this on a mechanistic level? So this is a very potentially complicated question, and to make things as simple as possible, we decided to use barrel cortex as a model of experience-dependent plasticity. Barrel cortex is a specific part of the primary somatosensory cortex that responds to whisker input. And many forms of experience-dependent plasticity have been studied in this system, and the mechanisms have been studied in a lot of detail. So therefore, after seeing whether the climbing fiber does anything at all to plasticity in the somatosensory cortex, we will be able to figure out how it's working. And more specifically, we decided to use this repetitive or rhythmic whisker stimulation protocol to induce plasticity in somatosensory cortex, and from there see if climbing fiber stimulation during this plasticity induction would change the plasticity phenomenon anyway. So this was a protocol that was based off of the HoltMat lab, who have studied the mechanisms underlying this phenomenon in detail, but essentially just rhythmically or repetitively stimulating whiskers at the frequency that mice naturally whisk at 8 hertz can cause pyramidal cells in the barrel cortex to potentiate their responses, meaning if you record from these neurons before and after plasticity induction, you'll see an increase in their response. And this was done in anesthetized animals. So applying that sort of concept to our experimental paradigm, I'm going to explain that here. So we're using a similar rhythmic whisker stimulation protocol to induce plasticity and look at the whisker-evoked responses before and after plasticity induction. So that looks something like this. So we will deliver air puffs to the whisker pad, or air puffs to the whiskers, and test the responses in S1 before a 5-minute period of rhythmic whisker stimulation, and then record the same neurons after to the same test pulses. And we're specifically going to try to look at the same population of neurons to see if their responses are different before and after plasticity. And how we do that is just by detecting whisker responsive neurons, or those that evoke responses within a specific window after stimulus onset. So here, for example, in the previous slide, I showed all the times I delivered the stimulus, and then detecting these events here. This cell would be identified as a whisker responsive cell, and we can test the amplitude of the responses before and after plasticity induction.
Question, Abby. No, so what's the time scale for the plot at the top?
This is a super long time scale. So each of these trials, this entire trial was 20 seconds long.
And
And
is
then
it like every second of stimulation?
no, so these are the stimulation is delivered once per 20 second trial.
So
So
then
this is a super long trace,
I see.
several
So
minutes
the
long.
the distance between each two dots is about 20 seconds.
Yeah. And then I have random inter-trial intervals as well. So this is just compiled from all my trials. This is not a
Okay.
continuous trace.
And then what lobule are you imaging from?
So this is imaging in barrel cortex.
Okay, we are now in
Yeah,
barrel cortex, not the cerebellum yet.
yeah,
Okay. And then the stimulation is by air puff.
yep.
Okay.
So this is what this would look like over an entire population. So an entire population of cells across several nights. If you, like, looking at the same 287 cells before and after plasticity induction, the population response looks like this before, and then this green trace is after. And you can see that we quantified here the area under the curve, which is kind of like a descriptive statistic, but really it's just the mean response um, in a specific window after stimulus onset. And you can see that it's higher.
And
So
this
that's,
is this induced after, am I saying this right? After about 5 minutes of
yes.
right.
Yeah. Okay. So now we know that this type of plasticity is dependent on, the whisker input. S1 plasticity is known to rely on the input signals. Now we're going to see if climbing fiber co-activation during plasticity induction is going to change this plasticity phenomenon at all. So this is our really basic experimental paradigm here. So this climbing fiber optogenetic stimulation in cruise 1 and 2 of the cerebellum, I'll explain why that's important in just a second, occurs during this plasticity induction period. And this isn't just some sort of willy-nilly stimulation. We're being really specific with the timing here. So on top of the 8 hertz whisker stimulation, we have 1 hertz climbing fiber stimulation. This climbing fiber stimulation at 1 hertz is similar to the spontaneous rate of climbing fiber activation in the cerebellum. And we are also timing each optogenetic stimulus such that it matches the natural timing of arrival of climbing fiber responses to whisker stimuli when they may convey an error. So what I mean by that is specific regions of the cerebellar cortex will naturally respond to whisker input in a specific context. That signal in cerebellum arrives at a peak of around 50 milliseconds after stimulus onset. So that's the period that we're going to optogenetically stimulate the climbing fibers to sort of artificially evoke an error signal in the cerebellar cortex.
So it was much less frequent in the previous slide. Why do you do now 8 hertz stimulation?
So this previous slide, these are the test pulses that are occurring like more than 20 seconds apart. And then this 5 minute, this is when the 8 hertz period occurs. And then these are just test pulses again after
I
plasticity.
guess then I didn't catch that. So the test pulses come once every 20 seconds, but
Yeah.
the actual pulsing happens at 8 hertz.
Correct, yeah.
So there's 8 hertz of pulsing for 5 minutes, and then there is once every 20 seconds of testing.
Yep. And then
Correct.
these are the responses to those test pulses.
Right. Now you understand. Thanks. And then here, what lobule are you now?
So here the optogenetic cerebellar stimulation is occurring in crus 1 and 2 of the cerebellar cortex. So this is where the stimulation occurs, and this is where the recording will occur. I don't know if you can see my mouse. I don't think you can.
No, maybe you use the laser pointer. Sometimes mouse doesn't show.
I'm not sure.
Actually, your mouse for a second appeared. I think, yeah, now we see your
Okay.
mouse. Yeah, it's good.
So this is crus 1 and 2 of cerebellum, which this is a sensory responsive region of the cerebellar cortex. So this will respond to sensory stimuli in some sort of error-driven mismatch sort of condition. And then this is where we're recording in somatosensory cortex.
One more methods question. So is this injection of channel rhodopsin in the olive, and then you have a cannula? Is that how you're doing?
Yeah, I have a couple of slides on that coming up. So yeah, I mean, the first, characterization we need to do is just make sure that we have our channel rhodopsin properly expressed in climbing fibers. So we have to inject it into the olive. There aren't any climbing fiber specific promoters. So you really need to target the olive directly. And that will give rise to climbing fibers terminating in the contralateral hemisphere of the cerebellar cortex. So here we have expression in the olive. This is like the highest density of expression is in the principal olive, and that primarily projects to crus 1 and 2. So this is like the specificity of our label. So the most of the expression I saw was in crus 1, a little bit in crus 2, and this was achieved with some really specific like angling of the pipette and specific volumes. So then this is what the labeling looks like in the cerebellar cortex against some Purkinje cells that are just stained post hoc with calbindin, so you can visualize. So we have our labeling in crus 1 and 2, and this is where we're going to be optogenetically stimulating now.
I see. So you're stimulating not the olive, but the climbing fibers.
Right. Yep. The terminals in the cerebellar cortex. So just kind of this diagram again in this context, we have Purkinje cells here. This is our calbindin staining, climbing fibers here, and then we also labeled excitatory terminals. So these are going to be like our granule cell inputs, which are giving rise to parallel fibers. And we just wanted to make sure that our labeling was specific to climbing fibers and not the granule cell layers. So the fact that you're not seeing blue down here means that we haven't gotten any granule cell labeling, but the fact that these blue terminals are overlapping with green here means that we have indeed labeled these excitatory terminals. And if you look really closely, you can kind of see that the path of the climbing fibers is nicely aligned with Purkinje cell dendrites. So we're pretty confident
Abby,
that
why did you not stimulate the olive directly?
So the olive directly was stained here. So this is the entire olive, and we also see that there's calbindin labeling in the olive as well. So these are neurons in the olive, and then the most of our expression was in the principal olive here.
Sorry, I think yeah, something else. I'm saying, why did you not put your fiber in the olive?
Oh, just because we wanted the specificity with the stimulation. So, I mean, if you try to excite the entire olive, you would definitely be getting climbing fibers that would go to other lobules, and also you would be directly activating the collaterals to the DCN. So the reason that we wanted to use blue light stimulation in the cerebellar cortex is that you would get specific activation of the climbing fiber to Purkinje cells, but it would be very unlikely that you're directly activating the DCN because the DCN is located far enough down that the blue light would not reach it directly. So that was our reasoning.
That's good.
Okay, so we have our labeling. So we wanted to make sure that also the way that we're stimulating is activating Purkinje cells in, you know, a somewhat at least functionally relevant way. This is the cell that would be reading out climbing fiber activity, and it's the sole output neuron of the cerebellar cortex. So it's really important to understand how the optogenetic stimulation of climbing fibers is actually modulating Purkinje cells. So this is a proof of principle experiment here where we just record Purkinje cell dendrites in Cruise 1 and 2. They're expressing GCAMP, and then we are also stimulating the climbing fibers at the same time. So this is what a field of view looks like, a 2-photon field of view looks like, looking at the Purkinje cell dendrites, and here's just some sample responses to the blue light stimulation. And by varying the stimulus parameters, we can achieve something that looks like a spontaneous event. So here we are using the light to evoke these calcium transients, and then you can see that the latency to the peak and the amplitude of this event was not significantly different from a spontaneous event. So now that we are confident in the specificity and of the labeling and the way that we're stimulating, now we can ask the question of whether optogenetic climbing fiber activation is modulating S1 plasticity. So because things get a little bit complicated here with this circuitry, I'll make it very, very simple and then kind of go back to some complexity later. So really the question here is, does this climbing fiber signal activation during plasticity induction modulate that plasticity in any way? That's the question. So how we're answering that is using 2-photon calcium imaging of S1 and climbing fiber stimulation in Cruise 1 and 2. And that climbing fiber stimulation is going to occur during rhythmic whisker stimulation. And then we just test in the same way these air puff responses before and after plasticity. So putting this all together, we have our GCAMP expression in S1. We have our climbing fiber expressing channel rhodopsin here in the opposite hemisphere. And then this is what our 2-photon field of view is going to look like. And we are going to be recording first from just excitatory and inhibitory neurons and seeing what their responses look like. So as you saw before, this is our baseline plasticity response. We see potentiation of excitatory pyramidal neurons. If the climbing fiber is activated during plasticity, also what we see is that this plasticity is sustained over time as long as the recording was occurring, and it's significant whether you're looking across cells or mice.
Abby, could you remind us? So it was 8 hertz stimulation of the whisker, and then you're doing climbing fiber stimulation at 1 hertz.
Yeah.
So some of these whisker stimulations co-occur with the climbing fiber stimulation.
Yep. So yeah, some of them will co-occur, and when they co-occur, the climbing fiber stimulation is delayed with respect to the whisker input.
Right. And then you do this for 5 minutes. And after that, you do the test. And now we're looking at the test results, which are only whisker stimulation.
Yep.
And we are seeing the activity in somatosensory cortex.
Yep. Exactly. So these are the responses in somatosensory cortex, the baseline. And now when we have the climbing fiber, we're going to call this rhythmic whisker stimulation plus CF. We see that this potentiation effect in pyramidal cells is no longer present. And it looks like this is not significant, maybe even some depression. And this potentiation over time is blocked, as well as the responses across all mice. So we essentially see that the potentiation of pyramidal cells that normally occurs is completely blocked by climbing fiber activity. In the same mice, we also looked at inhibitory interneurons. And so with the potentiation of pyramidal cells, what you normally see in a lot of more naturalistic forms of plasticity and learning is a concomitant depression of interneurons. And this kind of helps keep the overall balance of activity in S1. So that's what we saw. We saw a decrease in the activity of interneurons, which was significant across mice. They didn't really change over time. With climbing fiber coactivation, we also saw that this depression of inhibitory neuron activity was blocked by climbing fiber coactivation. So essentially what these results show is that the climbing fiber appears to be gating the plasticity or blocking that plasticity from happening. And we further split these interneurons into the populations of cells that are mostly comprising that interneuron population. So around 70% of these putative interneurons are going to be SST and PV positive cells in S1. And so we show that across all neurons, all trials, even ones that aren't even whisker responsive, we see that in SST and PV cells, we saw a decrease normally, and this effect was blocked by the climbing fiber coactivation. So across all cells and trials, again, what we see is an increase in the excitatory activity, a concomitant decrease in interneuron activity, which is represented in SST and PV cells, and all of that pyramidal cell potentiation is blocked by the climbing fiber. The SST and PV depression is also blocked by the climbing fiber. So we see these effects in the neural population. We also know that these populations are modulated by locomotion. So we wanted to perform an important control to show that these plasticity effects weren't confounded by any locomotion that was occurring during or after stimulus onset. So we show that we're going to just monitor the behavior. So we're going to monitor any whisker movements of the mouse. We're going to monitor any locomotion activity. And then we're also going to monitor the neural activity. And we see that the baseline plasticity effects are preserved in the absence of locomotion. So the mouse is still before and during and after stimulus onset. We still see the potentiation of pyramidal cells and depression of interneurons. And both of those effects are blocked by the cerebellar climbing fiber. And this isn't only just a control, but kind of an important finding that the cerebellum seems to be gating this plasticity in the absence of any sort of actual motor activity or motor behavior, which is one other example of potentially a purely sensory role of the cerebellum. Or in this specific context, this is kind of what this shows.
Question. So for this slide, did you say you focus on when on the apex where the mouse is not locomoting?
Correct, yes. So
Okay,
these are all the test pulse trials where the mouse is still during the test pulse, before and after the test pulse.
And then, so what's the, I'm trying to understand, because with whisker stimulation, we will be activating climbing fibers, perhaps.
Perhaps, yep.
I mean, I did periocular air puff stimulation, and that happens. So I guess we can generalize that to whiskers. So then this, so without the optical stimulation, without the artificial manipulation here, we know climbing fibers are going to be active, and despite that, we get the potentiation in somatosensory cortex. So is the idea here that if we increase this natural activation of climbing fibers, now we mess up with the potentiation?
I think the key here is that what we're doing with the optogenetic stimulation is providing like a synchronous activation of climbing fibers. So yes, climbing fibers naturally will respond sort of asynchronously to whisker stimulation or an air puff, but typically when an error or like some mismatch event occurs, you get this really synchronous activation of climbing fibers. And so it's kind of like a pathological level of synchrony that we're inducing with the optogenetic stimulation. There's really no way around that. But it is sort of, we can actually kind of use that to our advantage because the synchronous activation does occur naturally in error type contexts. So
So
I think that's the
if
key.
that's the hypothesis, there is an easy way to test it. In my life paper with Javier, when I was in his lab, so when I increased the intensity of the air puff, the synchrony increased. So potentially you can increase the air puff intensity to increase the synchrony and see if now that affects the potentiation without the presence of the optogenetics. How intense are your air puff stimuli? Do you remember the PSI?
Yeah, so they are quite gentle.
Yeah.
It is 8 psi or
Yeah, that does count as I did get pretty low synchronous activity at that. So do you think what I'm suggesting kind of
yeah, I think
makes
that's
sense?
absolutely true. Like there's a recent paper, Lena Williams and Holt Matt paper in 2025 that were doing this rhythmic whisker stimulation in vivo. They did a longer, like 10-minute protocol. And I think we were also, this is kind of offline, unpublished, but talking about if you very, very intensely, like aversively, repetitively stimulate the whisker, you'll get more depression. So yeah, that itself could kind of gate plasticity. Um, and I've kind of observed when I was troubleshooting this too that this rhythmic whisker stimulation does need to be quite gentle to get potentiation. Too high and it will not like the potentiation.
And what's the implications of that? That when there is a strong stimulus, whether in the form of an error or a strong sensory stimulus, now we don't want potentiation to happen in somatosensory cortex. Is it a little bit not counterintuitive because a strong stimulus or surprise or error should induce plasticity? And
Well, perhaps when the cerebellum itself is evaluating something as an error, those are conditions where you wouldn't want to induce plasticity in the cortex. So, I mean, I think if something is aversive and behaviorally you want to maybe not necessarily actively avoid it, but just not learn uh like a positive or any sort of association with this was when you would want to suppress activity. Um, but and I can speculate more on this
yeah.
too. But I think in this specific context, which is very, a pared back way of looking at this, we see that this is an error, like it blocks plasticity, but I think that the plasticity that could be induced or in the climbing fiber could be influencing could be bidirectional based on the state of other brain regions. And what the climbing fiber signal is actually conveying. So perhaps if it's conveying novelty or salience or reward, you could get the opposite effect or some other effect in cortex. So here could potentially be signaling some sort of error where you don't want plasticity to occur. But
Very cool.
okay, so we also just show this with a really like high level way of looking at this, which is receptive field plasticity using intrinsic signal imaging, which is kind of a technique that you can use to look at entire barrels in S1. So you see a spot get darker, and this is kind of a measure of like the hemodynamic response. And this is the region responsive to a single whisker. So before we were stimulating like a bunch of whiskers at the same time, but we saw that we still see this potentiation effect if we stimulate single whiskers when animals are anesthetized and we're able to manipulate just a one whisker at a time. And we see that the climbing fiber co-activation during this single whisker stimulation also blocks plasticity. And then a control condition of the climbing fiber alone also doesn't cause plasticity itself. So just another way of kind of confirming this general phenomenon that we were seeing earlier. And this point is not in the eLife publication that just hit the internet, but we also looked at what's the behavioral response to the air puffs. So normally when you puff the whiskers, the mouse will sometimes react to that and start running or start whisking. It's kind of like a surprise response you can think of. So I've split all of those trials into what I call go trials, where you see the stimulus or air puff onset, and then the mouse will start whisking or running. And I just quantify the overall proportion of trials where the animal just stays completely at rest, or where the animal actually starts locomoting. And we see that after the rhythmic whisker stimulation plus climbing fiber, the mice are overall less reactive to that stimulus as well. So they've seemed to be kind of like habituated or hypo-responsive to that whisker input. Whereas after rhythmic whisker stimulation, the mice continue to remain kind of reactive or responsive to the whisker. And we know that this isn't really confounding any of our other results, because we did look at things totally in rest. So the proportion of trials where the mouse are running doesn't really confound that overall plasticity result, but it kind of adds this point that it could be changing how the mice are perceiving the sensory stimulus. So if you depress plasticity, and the mice sort of become habituated to that stimulus, and now they're not reactive or moving as much after. That's kind of an interesting additional finding.
Question, Abby. Sorry, I guess I missed so what's the y-axis here?
So that's the proportion of trials where the mice are like the go trials. So the proportion of overall trials where the mice react to the stimulus.
And the react is measured as the mouse moving, doing something.
Yep. As the mouse like starting to run or starting to whisk.
Right.
Okay, so coming back to our central question of we've answered yes, we've seen several instances where the climbing fibers are indeed affecting plasticity outside the cerebellum. So the next point, how are they doing this? I'll kind of breeze through a lot of this because it gets kind of detailed, but I was really interested in the potential mechanisms of how this could be working. And so one hypothesis is that the climbing fiber could ultimately be recruiting or selectively modulating the interneuron population in S1. We know a lot about how these interneuron populations in S1 themselves gate plasticity, and so potentially the climbing fiber could be interacting with that local microcircuit to gate plasticity. So again, how is the climbing fiber maybe specifically modulating those populations? The first step that we have to show is it differentially affecting those populations at all? So now what we're doing is we're simply measuring the response to whisker stimulation, the acute response to whisker stimulation, with or without the climbing fiber co-activation. So this is just a simple test pulse with or without climbing fiber activation. And we see that across 53 animals, there's a slight decrease in the response of pyramidal cells overall, and a small increase overall in the entire population of interneurons. And then when you look more specifically at actual tagged populations of interneurons, here we are tagging in VIP Cre mice the VIP neurons and measuring their responses, similarly with SST and PV neurons. So there's a lot here, but essentially what we see overall is there's a definite increase in the response of SST neurons, a very small increase in the PV neurons, as well as a decrease in the response of VIP neurons.
Abby, I'm pretty confused. I'm sorry. How are these different from what you were showing us until now? Is this not the same protocol with climbing fiber and whisker co-stimulation? Why are we getting now different results?
So right here, what we're looking at is, so this is in the absence of any plasticity protocol. This is just simply like the basic test pulse responses. So we're kind of trying to investigate what might be happening during that rhythmic whisker stimulation period, how the climbing fibers might be modulating that.
How frequent is the stimulation for both climbing fiber?
So it's a 100 millisecond whisker test pulse with a 50 millisecond climbing fiber test pulse, and that climbing fiber test pulse is delayed with respect to the onset of the whisker.
And then how frequent? Like what's the interstimulus interval?
It is over 20 seconds. So we are looking at just these test pulses. So we're essentially looking at what the response is. So I said earlier, we had like 8 hertz whisker stimuli with 1 hertz climbing fiber. What we're looking at is what the responses in whiskers would look like during only those paired, like
right, before any prior 8 hertz thing. And then you're showing that with that one stimulation in 20 seconds, now the pyramidal neurons, there's a depression.
Yeah, there's a decrease in their response.
And when you pair it with climbing fibers, now it goes up.
In the interneurons. So looking at the interneuron subpopulations, we see an increase in the SST and PV cells. And then the VIP cells, if you look at the exact same VIP cells before and after, they don't look like they're being modulated. But actually, if you take a look at the overall population of VIP cells, there's a lot that are just completely turned off. So, we see 159 of them were responsive to the whisker stimulus. Only 132 were responsive to the co-stimulus, and the entire population itself was depressed in activity. So it appears that the VIP population, there are many that are just completely now unresponsive to the whisker stimulus that had previously been responsive, as well as the overall population, the amplitude of that activity is lower.
And I suppose you have all the controls to make sure this is actually climbing fiber stimulation, because could be anything else that the mouse sees the stimulus. And so this VIP change of activity is not a direct consequence of the climbing fibers. It could be just other stimuli that the mouse is perceiving at the same time.
Yeah, so we've looked at what does climbing fiber stimulation itself do. That doesn't do anything. It doesn't modulate the cortex at all. It requires the whisker stimulus to see any sort of modulation of activity. So we know that, this isn't like visual. We've also experimentally controlled for a lot of potential confounders. So, we've made sure that the mice can't see any light. We've also looked at trials where the mouse is not moving, rest trials, all of these things. Yeah. So the idea is, since we do see that the climbing fiber is capable of like acutely modulating activity in the cortex, it's possible that the climbing fiber is indeed selectively modulating these neurons to gate plasticity in S1. So the hypothesis would be that, and what we saw, as well as this kind of matches with previous literature, is that the activity of SST and PV neurons would normally decrease, which allows for potentiation of pyramidal neurons. So this is kind of the connectivity of the local microcircuit. You have SST cells connecting to the apical dendrites of pyramidal neurons, PV cells that synapse more on the soma. But if you decrease the activity, you release inhibition onto pyramidal cells and allow them to potentiate their activity. But if we saw the climbing fiber is increasing the activity of SST and PV cells, this could be a mechanism, and decreasing the activity of VIP cells, this could be a mechanism by which the inhibition onto pyramidal cells is increasing during rhythmic whisker stimulation, and therefore causing the pyramidal cell, depression or potentiation not to occur. So this is kind of what we think is happening during the rhythmic whisker stimulation, which then therefore would cause potentiation or a gating of plasticity with climbing fiber coactivation. So to actually test this hypothesis, we're going to do some experiments where we kind of test the necessity and sufficiency of these neurons to mediate the effects of the climbing fiber. So now that we know how the climbing fiber is acutely modulating the activity in S1, we can use DREAD manipulations to bidirectionally modulate the SST and PVs during plasticity induction to see if that's going to prevent the climbing fiber effect. So I'm going to walk through one of these experiments, and then there are multiple experiments that are kind of in the same logic. So the first experiment, we want to test whether SST neurons are sufficient to recapitulate the effects of the climbing fibers. So if climbing fibers are activating SST cells, is SST activation sufficient to block plasticity? That's kind of the question. So we locally express a activating DREAD into those SST neurons in S1, and then repeat the same experiment where now the SST neurons are going to be activated during the plasticity induction. So we see that you only need really like sparse expression of the DREAD in SST neurons in S1 to see these effects. And so we show that we activate the SSTs with the application of a DREAD agonist, and we show that with rhythmic whisker stimulation, if this occurs with rhythmic whisker stimulation, we block the plasticity effect. So this shows that SST activation is sufficient to capture the effects of the climbing fiber. Now the reverse experiment, if we were to suppress SST activity with climbing fiber activation, we should block the effect of the climbing fiber. So I'm going to kind of condense all these experiments into just these 3 panels because it kind of, makes it a little bit less confusing. So in this first experiment, I showed that SST activation sufficient to block plasticity. Now the opposite experiment, we're suppressing SST activity with climbing fiber coactivation, so the rhythmic whisker stimulation plus CF, and we see that we still get potentiation of pyramidal neurons. So now the climbing fiber effect is blocked. So this shows that SST neurons are themselves sufficient to block plasticity, and they're also required to mediate the effects of climbing fiber signaling. So hopefully that makes sense. And then because SST neurons don't get much input from outside the local microcircuit, we're going to go one step back and look at the VIP cells. So the VIP cells we know strongly innervate SST and PV neurons in S1 cortex. So we're going to repeat these same experiments now, but now our manipulations are going to be targeted to the VIP cells. So if you block VIP neurons, what you should be doing is, increasing the activity of SST and PVs to gate plasticity, and that's exactly what we see. So that looks like what the climbing fiber does. And then the opposite experiment, if you activate VIP cells, you block the effect of the climbing fiber. So again, this shows that VIP cells, their suppression is sufficient to capture the effects of the climbing fiber, and their activation or their suppression is also required to mediate the effects of the climbing fiber, because if you activate them, it reverses the climbing fiber effect. And I think the best evidence that we actually have that this sort of disinhibitory circuit between VIP, SST, and pyramidal cells, or even VIP to VV to pyramidal cells exists is in these experiments, we can not only look at pyramidal cells, we can look at the tag VIP neurons, and then the other population of cells should primarily be SST and PV cells. So we see that in the other population of neurons, so the interneurons, we see that we saw an increase in the interneuron activity and a decrease in the interneuron activity with the climbing fiber co-activation. So now this kind of like perfectly recapitulates the effect of the climbing fiber, and is showing specifically that it's through this sort of disinhibitory mechanism that this is occurring. So I hope that makes sense. So this is sufficient to like capture the effects of the climbing fiber activation on in the inhibitory population, and we get the suppression of inhibitory neurons even when the climbing fiber is active if the VIPs are activated.
What did you use to inhibit VIP?
This is dread activation or inhibition. So we performed some additional control experiments to kind of account for the fact that this manipulation was potentially could cause some like changes in excitability of the pyramidal neurons in S1, because we've lost sort of like some temporal specificity that we could have done if we had optogenetic stimulation itself. But we showed that even with those controls, that effect couldn't explain what we were seeing. So we are confident that this manipulation was enough to kind of show this effect.
Did you try to do auto?
I have, and I am currently doing some follow-up experiments. So, and I mean, it shows a similar effect. Yeah, it is more difficult to kind of like tune the timing perfectly to sort of like interrupt the climbing fiber events, because I didn't want to just blanket inhibit or excite them during the entire 5-minute period. But yeah, I mean, so far preliminary results suggest that it's similar. Okay. So then the final kind of leg of this project was to look at how the climbing fiber signals could ultimately propagate to S1. So I'll go over this pretty quickly too. But we're kind of like going back and expanding our complexity again. So we know that the climbing fiber signal has to go through the cerebellum, Purkinje cells to the deep cerebellar nucleus, but we don't really know where it could be propagating through the diencephalon. So we don't know like really what thalamic nucleus or subloop nucleus could be involved, but we know there has to be one more synapse somewhere in the diencephalon before S1. So this was kind of our hypothesis. POM and VPM are the main sensory thalamic nuclei that innervate S1, but the evidence that shows that cerebellar nuclei directly project to POM and VPM is pretty weak. And those direct projections are probably pretty weak. And so we were looking at another sort of intermediary nucleus that could be directly or like strongly modulating the activity of the sensory thalamic nuclei, or even directly modulating the activity of S1. And that's the zona incerta. So the zona incerta both directly projects to S1 and it projects to the higher order thalamic, the POM nucleus of the thalamus. And it's been previously shown that this POM thalamic nucleus is required for this pyramidal cell potentiation. Okay, so what we did is we just looked at the outputs of the zona incerta that receive cerebellar input, and we looked at where those projections were going. And we saw labeling in POM, we saw labeling in S1. So there's a lot of panels here, but essentially the takeaway is that indeed the zona incerta, and specifically the neurons that receive cerebellar input, are projecting to the POM thalamus and directly to S1 as well. So here's kind of a view of S1, the labeling in S1, as well as in POM. So the zona incerta seems like a plausible nucleus that could be modulating the activity of S1. Here's just another set of images. And this slide shows that there is indeed like a monosynaptic connection between the cerebellar nucleus to the zona incerta to the POM thalamus, which hadn't been previously shown, which is just kind of another layer of evidence that the zona incerta might be an intermediary nucleus. And also, the POM neurons that receive input from the zona incerta actually synapses onto VIP cells were visible. So if we saw earlier, these VIP cells are likely kind of a conduit of the activity from these thalamic nuclei. And so all that evidence together, and you can take a closer look too at the publication, suggests that the ZI might be involved in propagating this signal. And indeed, when we manipulate the populations of ZI neurons, so both the SST and PV neurons in the zona incerta, the SST ones being the ones that kind of directly project to S1 and the PV ones being the ones that directly to project to POM, we show that the basic transmission of the climbing fiber signal is blocked. So here we're just looking at the test pulses as before. So looking at whisker test pulses and climbing fiber coactivation. We saw that there's a suppressive effect on pyramidal cells and excitatory effect on the interneurons. And both of those effects were reversed by manipulating the zona incerta. And it seemed like specifically manipulating either the PV or the SST population had more selective effects on the excitatory or inhibitory population in cortex.
Are there any excitatory projections between zona incerta and thalamic or cortical areas?
I've never heard of that. I think the zona incerta is just essentially an inhibitory nucleus. So if there are excitatory projections from the zona incerta to elsewhere, they'd be very, very sparse.
And then it's now getting a little bit complicated because we have in one pathway, it's double inhibition going from zona incerta to the thalamus to the, I guess in both pathways, it's single inhibition, right?
Yeah. And so, it definitely is getting really complicated here in terms of like how the signal is actually propagating and even what is the relevant signal to be propagated, because a synchronous activation of the climbing fibers it's more likely to elicit like a biphasic response in the deep cerebellar nucleus. So meaning a synchronous activation of climbing fibers might cause this initial suppression in the deep cerebellar nucleus followed by rebound activation, whereas like asynchronous climbing fiber activity might only cause that initial depression of DCN activity. So we don't even really know, is that initial suppression or the rebound activation of the DCN the relevant output signal here? I would guess that, and I've really, if I modulate the intensity of that climbing fiber stimulus, I don't see any effects in cortex if it's not sufficiently strong to activate synchronous, activity of Purkinje cells. So I do think that the synchronous activation of Purkinje cells is necessary to induce effects outside of the cerebellar system. meaning that it might be this rebound activation phase that's actually the relevant output for at least for somatosensory cortex.
Or a very synchronized inhibition. That could be also like perhaps the inhibition is much stronger when there is synchronous. So it won't be equal as desynchronized climbing fibers, given that they all converge. So
yeah. And there's also like both excitatory and inhibitory projections from the DCN to the zona incerta. So, you know, that's understanding that signal propagation.
So DCN projections to zona incerta are not all excitatory?
No. There's actually a surprisingly large population of inhibitory DCN projections to the zona incerta.
This is interesting. I didn't know inhibitory projections go out of DCN to outside. I mean, I know there are inhibitory projections to the IO, but
yeah. So yeah, I mean, understanding that signal propagation would be, you know, a whole host of additional experiments. And, you know, each of these nuclei too, particularly zona incerta and POM, are going to be differentially modulated in different states.
What do we know from zona incerta's role with regards to the cerebellum?
We know that, and this is kind of just recent discoveries, like it may be that pathway, cerebellar DCN to the zona incerta, might be implicated in some social behaviors, also like anxiety-like behaviors, fleeing responses, feeding behavior even. And I think one thing that's kind of interesting too is the zona incerta to POM projection, which is a little bit more well characterized. We know that the zona incerta can be like during self whisking or self grooming versus like external sensory stimuli, they are differentially modulated. So one idea is that, you know, we see this gating of plasticity, but we could also see gating of activity during self move or self stimulation versus like external sensory stimulation, which you may during self stimulation not want to excite cortex as broadly as during like a more salient relevant like external sensory stimulus.
And you're saying we get zona incerta's activation more during self stimulation?
Correct. So like more activation and meaning more inhibition onto the POM.
Yeah.
Yeah. And how the cerebellum could be involved in that or propagating signal, you know, multiple ways of like of doing the math and like counting the number of inhibitory synapses, right? Like
although like optical stimulation is not self-stimulation, so
right. It's completely, you know, it's essentially, you know, we're doing our best to try to convey something relevant, but it's not, it's just artificial. Okay, so to kind of wrap up, my little symbols aren't showing up on this slide, but I've shown you a potential context where it may be conveying some sort of error. So like maybe in the somatosensory cortex, reinforcing plasticity based on an errorful input risks encoding an unstable or inaccurate representation. So in essence, you wouldn't want cortical circuits to learn from a signal that the cerebellum itself is labeling as wrong. And that might be what we are conveying with our optogenetic CF stimulation. But what if the climbing fiber is naturally being recruited in other things we know that recruits it? So like reward, novelty, salience, the activity of other brain regions, as well as, you know, is going to ultimately modulate the effect in S1. So we could be seeing different results in depending on what the climbing fiber might be conveying. But really what this study shows is the climbing fiber has the possibility of conveying something to S1, you know, and we kind of like tease apart a potential mechanism by which this could occur. But all of the the relevance of this in other contexts is to be explored. And really just the complexity of this circuit kind of doesn't detract from our potential pathway that I've suggested. It's just one potential pathway, but all these other regions, I mean, like the feedback connections, how they're all interacting. So this slide is really just to show that the complexity of how the cerebellum can modulate activity outside, and specifically how the climbing fiber could be mediating these effects can be, I think there's large implications for that. So yeah, I don't think the slide came up either, but that's essentially my summary. I'm wondering if you have, oh, sorry, before that, I have my conclusion slide. So what we've shown is in support of existing evidence, the cerebellum may have a more sensory role. It's not just restricted to motor control. And our new and important findings are that the cerebellar climbing fibers can convey error signals or some sort of instructive signal to the neocortex, and that the cerebellum controls the inhibitory network in the neocortex to gate this plasticity. So I'd like to acknowledge my advisor, Christian, and my committee, Anne-Marie Oswald in particular, my chair, who kind of steered me in the direction of looking at interneurons and cortex, which I really enjoyed doing. So with that, I'll take any questions if there are any.
Yeah,
Sorry, we're over time.
that was really nice. Yeah, thanks. We did have some people who left, I guess, at 1. Very nice. I think what we need now would be nice to do the more direct, like cerebellum to zona inserta, like the actual pathways to be more certain that the effects of these interneurons are actually coming from climbing fiber activation, or recordings from the DCN, like recording from these nodes along the way. But it's very, very insightful piece of work.
Yeah, I'm super interested in like really understanding, and in particular too, what I found kind of interesting is that all of the nodes that I looked at have neurons that express SST. So like, and calbindin. So like in inferior olive, you can label with calbindin. Obviously the Purkinje cells, the SST neurons in the zona incerta, and the SST neurons in the somatosensory cortex, which are ultimately mediating this effect. Plus, these SST neurons in cortex actually are known to respond to the similar things that the climbing fiber is activated in. So like unexpected events, salience. So yeah, that whole inhibitory network of somatostatin and calbindin expressing cells is pretty interesting.
And the circuit model here is that you did mention perhaps inhibition followed by rebound, but with the inhibition of the DCN, we get, I guess, activation of zona incerta, which leads to inhibition of the cortex, which leads to disinhibition of VI, which means inhibition of VIP neuron. Like it's very common, but I'm trying to see there are like 3 negative signs there, and that's how we get activation of SST, and then perhaps prevention of plasticity.
Yeah, essentially, like, so climbing fibers, activate Purkinje cells, gonna suppress DCN initially, and then DCN suppression will.
I guess that's the key. Does it inhibit or activate zona incerta? Because we did
yeah, that's the question. Like, is it that initial DCN phase initially suppressing or activating the zona incerta because it has those bidirectional, or like those 2 inputs? But sort of the initial way I was thinking about it is actually this rebound activation of DCN is going to activate zona incerta. The zona incerta activation suppresses POM activity, and POM is the node we know is like required to be involved in this form of plasticity. So suppress the POM activity, and then suppresses VIPs.
Because POM
POM directly projects to VIPs.
And now VIP is suppressed and SST is activated, and now we get this prevention, well, basically the gating of plasticity.
Yeah.
Okay. Very cool.
And alternatively, if you're convinced that it's not the rebound activation, maybe it's just DCN is suppressed. DCN then like suppresses zona incerta.
Yeah.
Which would release inhibition directly onto S1
Yeah.
VIP cells.
This is the complexity with systems neuroscience, that we do go there and manipulate one pathway, but then like you're saying, DCN does both excitatory and inhibitory outputs. Yeah. I mean, doing pathway-specific manipulations help. At least we know what pathway, like, are we manipulating the inhibitory inputs to zona incerta or the excitatory? But yeah, thanks a lot, Abby. It's 1:15. We should.
Yeah, thank you so much for your time, and I'm sorry I went over.
Sure. Good luck with medical school.
Thank you.
Best.
Bye,
Bye.
everyone.
Bye, everyone. Thanks. Bye.